All right, so today we're going to keep talking about inheritance and gene expression. So how do phenotypes match the genotype? So just before we get into that, let's do a quick review, just to make sure we're all on the same page before we start digging in. So remember that what we're talking about here is inheritance. So the DNA that you get from your biological parents. So for every single gene, you inherit one allele from your mother, and you inherit one allele from your father. Now remember that when we're talking about alleles, we're talking about variations of a gene. So for example, if the gene that we were talking about codes for eye color, the alleles would be like blue, green, brown. If hair color, let's say brown, blonde, red, those would be our alleles. If we were talking about height as the trait, the alleles would be short or tall, right? So together, those two alleles, remember you inherit two, right? One from your mother, one from your father. Together, those make up what we call your genotype, right? So those are the genetic copies of the alleles that you inherit. What that actually looks like, so when we're talking about how something is expressed, what we mean is what it looks like. Right? So how does that actually manifest in a person? So the word that we use when we're talking about that is phenotype. Phenotype being what the genotype actually looks like. So we talked about dominant and recessive. So dominant being the alleles that will always be expressed. Recessive can only be expressed if they are by themselves, right? So if a dominant is expressed, the recessive is there, but we won't see it. So alleles that are dominant are written with capital letters, so like an A. Alleles that are recessive are written with lowercase letters, so like an A. So the capitalization of our letters tells us whether the trait is dominant or recessive, and dominant alleles will always be expressed. So what we've been looking at so far would be like if we had red and white, we would do a Punnett square to see what the possible phenotypes are, right? So we have RR being crossed with RR. All of our offspring look the same here. And because this is dominant, this would be 100% red, right? All of our offspring would be red. So what we want to look at today is what happens if neither trait is recessive or dominant? Like, how can that affect how that looks? Because not every gene has a dominant and a recessive allele, right? Not every combination works that simply. And so today we're going to look at what happens when neither are dominant or recessive. So this time, instead of red being dominant, so it's still going to be two capitals, but white here is not recessive. White here is also going to be dominant. So the way that we show that is that we actually use capital letters for both. Because if we were to use the lowercase, it would imply that the white is recessive, and that's not the case here. So we're using capital letters for both. And so there are two different scenarios here. So these two cases both have the same, so if we were to do a Punnett square for this, we have red and white being combined, right? So all of these are going to be RWs. And we just kind of write those in alphabetical order because we don't have a dominant recessive here. So notice that this is 100% RW. So these two offspring actually have the same genotype. The difference is in how they are expressed. So different alleles have different ways of being expressed. So we're going to talk about the two differences. So if we look at them, what we see is that this one is kind of like a patchwork, right? You've got strips of white and you've got strips of red. So we're actually seeing both of those genes there. Whereas over here, we see kind of the blending with what happens when you mix red and white, right? So we're going to dig into that a little bit here. So the first one that we're talking about is incomplete dominance. So this one over here. So in incomplete dominance, neither allele is completely dominant. So what that means is they're kind of on an equal playing field. And so what happens there is they're kind of like friends. They work together and they meet somewhere in the middle. So with these, for a heterozygous, meaning that means different, right? So that'd be like RW. if it's heterozygous, we see a mix or a blending of the two. So if you have red and white, we get pink. If you had, let's say, like tall and short, you would get like medium height. So it's kind of like they're meeting in the middle. I'll add that in there. So it's important to note here that when we're talking about this incomplete and this codominance, the inheritance pattern is the same. We can still determine the probabilities by using that Punnett square, right? The difference here is in how that genotype is expressed. That's the only thing that's changing here. So some examples of that is really common in flowers. Okay, so when we have red and white flowers breeding, red being, so if you are red in this case, you are homozygous dominant, and white is also homozygous dominant. When they come together, they make pink flowers. And so the pink is actually the heterozygous form here because it's in the middle, in between red and white. So when you see that, we see that in-between state, that is incomplete dominance. Neither gene is taking over. So another example is the Andalusian chickens. Oh, these are actually really cool because their forms, they have black, that homozygous dominant, and white. The heterozygous, so the BW, so the heterozygous form, remember, because this is incomplete dominance, we know it's incomplete dominance, it should be meeting somewhere in the middle. So traditionally we would expect that a blue and a white make gray, right? That's pretty common. But in Andalusian chickens, their heterozygous state is actually blue, which is kind of funny. So when those two genes meet, their middle ground is in kind of a bluish tinge chicken. So they're actually very like wanted in the farming industry because they're very rare. So those are incomplete dominance, right? They meet in the middle. The second one, and I'm just going to go back to this original drawing here to show you this, is codominance. So co means like you're existing together. Like if you think of like a co-op, right? People are living together. Or in co-ed dorms, right? It's men and women living on the same floor. So you're living together. And so what we get in codominance, if you look at this picture, is we actually see both of them. So neither are taking over. They're actually coexisting, living together. So that's one way that I like to think, like to help to remember it. When I see that co, I think of coexist, right? Meaning like they're living together. So with codominance, they're both being expressed at the same time. So we can actually see both of those original phenotypes in the offspring. So the heterozygous phenotype will have both phenotypes being visible. So if we had like black and white, we get spotted. So we see that in these Aramonite chickens. So if we had a black chicken and a white chicken, so here, remember, black would be homozygous dominant and white is homozygous dominant. And when we cross those, oops. We see this heterozygous. And that's what we see in the offspring. We actually see, so you see the Bs and you see the whites, right? You see both showing up in the offspring. And these are, once again, in the farming industry, we've got like special names for all of these. So Aramonite chickens are chickens that show both colorations. And when you see that, it shows that heterozygous nature, right? We've got both genes being expressed. So cattle is another one, and this also shows up in dogs all the time, where when you have a red crossed with a white, the offspring are what we call like speckled or spotted. And they actually have a special name for that coloration pattern. So roan is that special coloration pattern. And you see roan, we're actually going to see roan again in the next example. And it's just talking about that coloration where you have both light and dark, right? Light and dark. And what that shows us is that red-white, that heterozygous nature. So same with, we have this horse here, right? We see that same coloration pattern with the red and the white. And we see that here. So this should actually be not be lowercase because it's not or recessive. And so one thing I just wanted to also remind you of is, like you see here, we've talked a lot about when two homozygous dominant things cross. Codominance is not restricted to homozygous dominant things. So here, we could take this roan cow, this RW, and cross it with anything, right? You could cross it with another RW. That doesn't change the coloration pattern. It just changes the probabilities for its offspring, right? So before, it was like all of your offspring are going to be this heterozygous state. If we cross those, now you've got 25% are going to be red, 50% would be roan, and 25% would be white. And remember, these are probabilities, right? They're not for sure. This is predicting. If we were to predict what their offspring would look like, that would be our prediction. And so same thing with any of these, right? So if we go back to this, to these Andalusians, this incomplete dominance, we could take a Andalusian chicken and cross it with another Andalusian chicken. Because remember, Andalusian chicken's just describing the coloration pattern. And we would see kind of the same distribution, right? So there's 25% chance of having a black. This would show 50% chance of having another blue chicken. And then 25% of being white. Okay, so this whole thing, it doesn't change what we were talking about before when we were talking about Punnett squares. It just kind of adds another factor to it. So you have 2 worksheets this week, so take a look and make sure you know which one you're looking at. Because whether the inheritance pattern is incomplete or codominant, that's going to change what your potential phenotypes look like.